Hello students, this is chapter number 10, Microbes in Human Welfare. This is video number 2 and in continuation. Today we are going to study microbes in sewage treatment. Now, what is sewage? We all know that sewage, that is the municipal wastewater that contains large amount of organic matter and microbes. And this sewage is treated in sewage treatment plants to make it less polluted. The sewage treatment includes two stages. One is primary treatment and another is secondary treatment. Talking about primary treatment first, this primary treatment, it involves removal of particles physically. It includes two things, two steps: removal of floating debris by sequential filtration, and second, sedimentation, where the water is allowed to settle, and in the sedimentation tank where the soil and small pebbles settle down, and then the clear water which is on the top, which is known as the effluent, is removed. The supernatant, that's the clear water, which is known as primary effluent, is removed, and the soil and the pebbles which settle down, the solid which settle down, is known as the primary sludge. Now, next comes the secondary or biological treatment. This primary effluent, that is the supernatant, that is passed into large aeration tank. You can see this is the aeration tank. This primary effluent is coming in the aeration tank here, and then it is constantly agitated. Now, because of this agitation, it allows vigorous growth of useful aerobic microbes. Yeah, because agitation taking place over here in this aeration tank, aerobic microbes increase in number, which is known as flocs. Flocs are bacteria associated with fungal filament to form a mesh-like structure. These microbes consume, because they're aerobic microbe, they consume the organic matter in presence of oxygen, and thus it reduces the BOD, the biological oxygen demand of the effluent is reduced as the microbes grow in number. Biological oxygen demand, what is it? Let's understand that. It is the amount of oxygen consumed by the bacteria to oxidize all the organic matter in 1 liter of water. BOD is a measure of organic matter present in the water. If organic matter is more, the water is polluted. And as the BOD, greater is the BOD, greater water is more polluted. And as the BOD reduces, that means the water is less polluted. Now you can see from the aeration tank, the effluent is then passed into a settling tank. You can see over here. This is the aeration tank, and then the water comes into the settling tank. And in the settling tank, the bacterial flocs, they undergo sediment. They are collected at the down over here. This sediment is known as activated sludge. A small part of this activated sludge is pumped back into the aeration tank. You can see the line over here. It is being pumped back into the aeration tank, and this acts as an inoculum. The remaining sludge is pumped into large tank, which is called anaerobic sludge digester. The remaining of that water is pumped into anaerobic sludge digester, where some anaerobic bacteria are present, which digest the bacteria, fungi in the sludge and produces gases like methane, H2S, CO2. Together, it's known as what? Biogas. Now this water is quite clean and this effluent is released into the natural water bodies like rivers and streams. So you can see the overall steps over here also. Primary effluent comes into aeration tank, constant agitation in the aeration tank, then aerobic bacteria in the flock consume the organic matter, BOD reduces. After this, it goes into the settling tank. Flock comes in the aeration tank, then it's anaerobic sludge digester. This anaerobic sludge digester digests the anaerobic bacteria, it produces the biogas, and then this water goes into natural water bodies. So this is how the sewage water is treated and put back into the natural water bodies. You can see how dirty we make the water. Just see, in name of rituals and all, we put so many things in it. The Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plan to save the water body from water pollution. Now, children, the next we are going to study the microbes in production of biogas. What are the microbes required in the production of biogas? Biogas is a mixture of gases that we have just now studied. It's a mixture of methane, carbon dioxide, hydrogen, hydrogen sulfide. But mainly the gas is methane. And this biogas is also produced by microbes such as methanogens. Methanogens are microbes which are anaerobic microbes, and they grow on cellulosic material, and they digest the cellulosic material. Why? Because cellulosic material is rich in carbon, and thus they produce methane, carbon dioxide, hydrogen, hydrogen sulfide. These methanogens are also known as methanobacteria. Methanobacteria are found in the rumen of cattle. We all know that biogas is used for cooking and lighting. Cattle dung or gobar, that's commonly known as, contain these microbes. So the dung is used to produce biogas, the gobar gas, also known as gobar gas. Now let's see the structure of a biogas plant. The structure of a biogas plant first contains a concrete tank. This tank collects the bio-waste and the slurry of the dung. It is present inside the underground, I would say present underground. A floating cover is placed over it. You can see over here, this is the floating cover which is placed over the digester. And when the biogas gets filled inside it, it rises up. There is an inlet tank. This is the inlet tank where we put slurry and mixed with water. And there is an outlet tank to remove the spent slurry, which is a very good fertilizer. And lastly, you can see this pipe. This is an outlet of supply of biogas. IARI, that is Indian Agricultural Research Institute, and KVIC, that is Khadi and Village Industry Commission, they developed technology of biogas production in India. Now the next topic is microbes as biocontrol agents. There are many microbes that can be used as biocontrol agents. This is a way of controlling the plant diseases and pests. For example, ladybird, that is beetle, you can see over here. This is the ladybird. Controls aphids, dragonfly, and dragonfly controls mosquitoes. See, generally what we do is to kill the pests, we use pesticides, chemical pesticides or insecticides, but both are very, very harmful to the organism as it causes pollution, it causes many diseases. You can see over here, a child born with such a big head. So biocontrol method is has no such problem. So we can switch over to biocontrol agents. The first is Bacillus thuringiensis, or BT, commonly known as BT. It controls the caterpillar of butterfly. You take dried spores of BT, which is available in the sachets, and you mix it with water and spray it on the vulnerable plant, such as brassica and fruit trees. Now, these spores are eaten by the caterpillars, and when this BT or this bacteria, this Bacillus thuringiensis, goes into the gut of the caterpillar, a toxin is released that kills the larvae. Now, scientists have introduced Bacillus thuringiensis toxin genes in the plants like BT cotton. You'll study more in chapter number 11. Next is Trichoderma, which is a fungus. It's seen in the root ecosystem and it controls several plant pathogens. Third is Bacallovirus. This Bacallovirus, it belongs to the genus Nucleopolyhedrovirus. They attack insect and arthropods. These Bacalloviruses are species specific. They are narrow spectrum insecticidal applications and integrated pest management program to conserve beneficial insect. This comes under IPM because it's an integrated pest management program. It does not, these viruses do not kill other insects which are useful to us. The last topic, that is microbes as biofertilizers. Microbes can also be used as biofertilizers to enrich the nutrient quality of the soil. For example, bacteria, fungi, cyanobacteria. Rhizobium, which is a symbiotic bacteria found in the root nodules of leguminous plants, and free-living bacteria in the soil like Azospirillum and Azobacter. They are some of the microbes which can be used as biofertilizers, which increase the nitrogen content of the soil. Then mycorrhiza. This can also be used as a biofertilizer. There is a symbiotic association of fungi, for example, glomus, with the plants. This fungus gets food from the plant, and in turn, it absorbs phosphorus from the soil and passes it to the plant, gives resistance to root-borne pathogens and tolerance to salinity and drought, and give overall increase in plant growth and development. Then one more biofertilizer, that is cyanobacteria or blue-green algae. They are autotrophic microbes, as the name itself is saying, blue-green. They fix atmospheric nitrogen. Examples of cyanobacteria are Anabaena, nostoc, oscillatoria. In paddy plants, rice plants, cyanobacteria serves as a biofertilizer. It also adds organic matter to the soil and increases its fertility. So with this, we finish with the chapter. I hope so the things are clear to you. Thank you.